Hello everyone. My name is Nagarjun. I'm going to talk today about the analytical characterization techniques for nanoparticle mRNA therapeutics. So I just put this slide, um, this image on here. So just showing the complexity of a small molecule, and then as you go to a antibody drug conjugate, a peptide, a recombinant protein, and once you have a map, and then you go for a viral particle and a cell. So the complexity of biologicals keep on increasing. So for today, I'm going to start with like a confidentiality statement. So right now, whatever I'm going to talk with you guys is kind of like mostly is the literature which I gathered, and it's mostly my personal opinion. Nothing related to any confidential data has been presented here. So the overview of the talk for today would be like to introduce about the mRNA therapeutics and cell and gene therapy products, and really talk about like analytical method development in drug product life cycle. And I would also like to introduce like critical quality attributes, and I would also love to talk about like the analytical methods to measure these critical quality attributes. And these are more focused towards the mRNA therapeutics. And I'll give my final thoughts on those. So just looking at the brief therapeutic landscape of an mRNA therapeutic, so ideally you can look at like it has a very long history of like, it started in 1961, and in 2020 we have our first mRNA vaccines that are being like used to really reduce the COVID pandemic. So, but like really looking at how these nanoparticles and mRNA have been like applied across. So the major breakthroughs came in like 1989, where they tried to use a cationic lipid mediated mRNA delivery. And at the same time, they also tried in 1978 is the liposome entrapped mRNA delivery. And as all these research work towards, at the same time, work more towards the stabilization of mRNA and really making mRNA a more stable molecule that can like incorporate into delivery vehicles. And as you go into the 2000s, where CRISPR-Cas came in, and then ideally people have tried mRNA vaccines for cancer and infectious diseases, but the COVID has been approved, and right now in 2022 there are like a lot of like mRNA-based therapeutics have been like applied across different diseases. So when we go ahead and look at this cell and gene therapy products at the same time with an mRNA vaccine products, so there are kind of like more than like it used to be very small number like in 2 years ago, but right now we have a lot more products that are being approved in the cell and gene therapy space. Primarily like with the gene editing, the Cas-GUL, which has been recently approved to treat sickle cell disease as well as beta thalassemia. And there are other gene therapy products which are like Luxturna for the retinal disorder, and uh, for the hemophilia, and Zolgensma for like SMA. And at the same time, there is this CAR-T products which are very specific, Kymriah for the leukemia and lymphoma. And talking about more, as this talk is more targeted towards this mRNA vaccine, so there are two mRNA therapeutics. So there are two mRNA vaccines that have been approved: the Spikevax for COVID-19 and the commentary. So for COVID-19, and the prediction is that like by 2030 there'll be like 30 to 60 gene therapy products, and really looking to treat around more than 3,500 uh, patients. So this is all good science, but like to really understand better from a drug development life cycle and analytical development. So I just want to introduce like, when a drug product life cycle really happens. So it really happens based on a clinical trial. You do a preclinical, you collect all the data, phase 1, phase 2 for healthy, and phase 3 approval and market. And when you look at the same cycle from a product development cycle, you say like an early stage development, which ideally includes your preclinical phase 1, and late stage really includes your phase 2, phase 3, and commercial is kind of more like you approval and you go to the market. And looking at the same cycle from a manufacturing point of view, you have a research and development, and then you do have your product go into an engineering runs, and then you have your GMP manufacturing, and then you go into commercial manufacturing. So on same lines, when you look at the same cycle from an analytical development point of view, you kind of have like, you define, design, evaluate, and optimize a method. You qualify a method, validate a method, and then verify a method. And which is again, parallelly, qualification is done around the phase 1, phase 2. Validation is like phase 3, and more verification, the approval, and the market phase. So for mRNA therapeutics, you put like the cycle into a manufacturing scale. So the primary steps would be like have a really research and development. You create this linear pDNA, and you kind of like manufacture this bulk mRNA, and then you kind of create this delivery molecule with the lipid nanoparticle and mRNA, and your final drug product should be like able to good to be go, and you should be able to characterize it. So what are the regulatory guidance for this mRNA therapeutics that are available right now? So from an agency-based regulatory guidance right now, look at EMA, we have like a guidance paper that recently came out, which talks about the quality aspects of an mRNA vaccines. And even FDA had produced, in 2018, they produced like a liposome drug product, like how we talk about like the pharmacokinetics and the CMC. And they recently came out in April 2022 with the biologicals, which kind of like they kind of include them into like nanoparticle guidance, as you really need to understand these mRNA therapeutics in a very critical way. So they kind of like give guidance from a nanoparticle. On the same lines, there are different articles that have been produced regarding from non-agency based methods. So these are some of these examples from USP. You have like an analytical procedures for mRNA vaccine quality, and the biopharma has like clearly defined some of these critical quality attributes for like an mRNA therapeutic, which primarily mRNA is being encapsulated in a lipid nanoparticle. So as we understand these guidances, we also need to understand the bottlenecks. So the bottlenecks for the mRNA LNP manufacturing really includes like so as I already showed this schematic where you have a linear pDNA, plasmid DNA, where you kind of generate your bulk mRNA. And once you have your bulk mRNA, you're going to mix with your lipids and to make your LNP with mRNA. You have this lipid nanoparticle, and then you have your final drug product. So the key areas R&D CMC has focused to really meet the commercial demand and improve safety was kind of like one of the biggest challenges. Like RNA scale up challenges were always there. And again, the product potency was also suboptimal. So sometimes you might need to have like higher doses to get like efficacy. And the other thing is like when there is kind of this challenge of like analytics available to really define this product in the right way, and really defining a critical quality attributes to define that final quality of the product is always a challenge. And for this specific talk, I'm going to really focus on the suitable analytics and like properly define like critical quality attributes, and what kind of attributes and what are the methods that are applied for these lipid nanoparticles. So before I talk about those, I would like to talk about like these critical quality attributes. What are these and how FDA defined them? So from the lens of an FDA guidance for critical quality attributes, a critical quality attribute is an attribute that is within an appropriate limit, range, or a distribution to ensure the desired product quality, to define a product quality. So this is a very specific cycle, like how these critical quality attributes for each product have been defined. So to start off with, like when you have like a final product, like when you have like a drug product, like you want to define the critical quality attributes, the first things you ideally set up is like a quality target product profile, which is like called like QTTP. And we define that by different approaches. So one would be you understand the mechanism of action, you understand the efficacy and safety, uh, dosage form, like you get all this information from your preclinical, and again route of administration, and desired quality attributes, and like and regulatory requirements, as this product has to go, uh, what are the requirements for this specific, what is the regulatory guidance available. Once you define all those things, you come up with a list of quality attributes, and you give them kind of scores based on the criticality, and you rank them, in such a way that like which gets the highest score will become a very critical quality attribute which needs to be really understood from the product itself. So once you define those critical quality attributes, and it may be like you might not have more information about that quality attribute from the product itself, so that puts it in a higher risk. So you kind of like understand all these uh, factors, and then you kind of define a control strategy. So one example of a control strategy is like, let's consider you have a product, and it works at a specific um, formulation buffer pH or salts, and you want to really check like what is that, if I change any of these attributes, how does this product really behave. So you go ahead and do some of these control strategy studies, and really define, understand the product. Is there a big change in the product, or like it's not really affecting the product? And once you understand these, this is where you kind of define your control strategy, and then you go back to your process and say like, oh, if the process deviations are like happening in this range with our control strategy, the product should be like still good to go in a manufacturing point. And once you kind of have your control strategy defined, you go back and do your risk assessments. So the way we do risk assessments to improve the process, kind of like ideally you look at like literature. Again, as you develop this product itself, you have the platform knowledge, and then you understand the product understanding, and then you go ahead like understand the process itself, like what is your, how is your raw materials, what are other things from the process, like you gain all that knowledge, and you do a repetitive process of this to really define those final critical quality attributes. Once you identify the list, you kind of do all these studies, control studies, and you kind of define the operating parameters of each attribute, and then you kind of like define that. So on the same lines, like so so this is from the FDA guidance, like how a critical quality attribute should be like identified. So based on this, we kind of like have some initial knowledge of like what are the critical quality attributes that are really required for an mRNA LNP manufacturing scale. So, but to really define that, like, and lipid nanoparticle has critical components. So the critical components that a lipid nanoparticle will have is an ionizable lipid, a cholesterol, a helper phospholipid, a pegylated lipid, and mRNA cargo. So ideally, when you look at like this is the mRNA cargo, and the rest of these particles are like the different lipids. Again, so ionizable lipid plays a role in really holding kind of your mRNA and increase the efficiency of delivery. Cholesterol, helper phospholipid, pegylated lipid, these all play a role in really helping, form that particle itself and fluidity, as well as this colloidal structure. That's what they really do. And mRNA cargo, it all depends on like what's the size of the mRNA you want to, how many mRNA you want to put in. That's a completely different area where we can discuss more. So based on this lipid nanoparticle mRNA lipid nanoparticle, the critical quality attributes can be like defined as like you can say like we want to understand the strength of this particle. We want to really define the compendial testing, and then when you manufacture these, the product related impurities, like, these are all raw material coming from. Each raw material has to be like any excipients, we need to understand them. And safety, what is the dose, what is the how does the size play a role? And the critical thing would be potency, if this batch is produced, and what is the potency of this batch, is it viable for to do any studies? And purity is also important. You always want to have like the highest purity of your product. You don't want to have any impurities. And a process related impurities also play a role, which is kind of like when you're manufacturing it, like you are really pushing different aspects. The product is kind of coming from the raw material, whereas the process itself, like you will be introducing different buffers. You want to really understand how this process related impurities are going to really play a role. And identity. So identity also is kind of, am I really producing the right kind of like a lipid nanoparticle? Is it what I am anticipated to see, and can I use this? So right now, like, so these are kind of like defined as critical quality attributes for the mRNA LNP manufacturing. So based on this, we can actually go ahead and individually look at like each critical quality attribute, and we can try to understand what are the analytical methods that are applied for an mRNA therapeutic based on each quality attribute. So we can start off with like identity and purity. So the attribute identity ideally comes from two places, right? One would be a sequence identity, which is like an mRNA. So you have an mRNA, you just want to be sure that like whatever mRNA are you like packaging is the same sequence identity. So it's very important in such a way that like you don't have changes or mutations in the sequence. So you ideally use like a reverse transcriptase PCR or like a next-gen sequencing. So a very important thing to understand, a release assay is kind of an assay where you kind of like do a lot of like qualification and validation, and really understand the limits of the assay, and really define like a limit of quantification, and you do a very thorough, like the assay has to be established very thoroughly in such a way that like you always have your system suits running, and when you run these assays, like you are very confident with the data that comes out. So those are kind of release assays. They are very stringent, and that's the way we need to develop these assays, because they are really talking about a very specific quality attribute, and that attribute has to be really defined from that assay. So lipid ID, so it's kind of like confirm the ID to appropriate lipids. So normally these are like through liquid chromatography methods. Ideally you use like an ELST method or a charge-based identification, which is the CAD method, or you can also go use like a mass spectroscopy method. And these methods are very specific for each lipid. So ideally you have to have a control lipid to run as your control and really identify it. So when it comes to purity, you really look at like, mRNA capping, because it really provides the translational efficiency and the RNA stability. So you really need to understand that 5' capping. And then you also understand the poly A, which is makes the stability and the translational efficiency. So normally the capping is understood through an LC-MS method, whereas the poly A is through a qPCR or a ddPCR method. The length and so also the length and the distribution. So normally when these kind of like transcriptions happen, the length can actually extend, or like you will identify like a specific length on this mRNA which is like more stable. So you need to identify that through an LC-MS method. How much, again, it also plays a role. Like if you have a longer length, the packaging of this mRNA into the LNP might not be really suitable. So another thing we also look at is the RNA integrity, which is like the full length. Is it like, as I said, it's like a full length, or like is having that extra length, or a shorter length, which can also like, is it degrading, it's forming fragments which can become impurities to, these are things we'll also look at through a capillary electrophoresis, or kind of like a regular phage electrophoresis or liquid chromatography. And these are all like release assays, so they have to be pretty critical. So looking at the next attribute would be potency and strength. So potency is ideally defined as kind more like, you have a drug product and drug substance. So drug product is kind of your final, you want to understand what's the minimum dose, like, that will give you that efficacy, that's what like the potency is defined for a drug product. When it comes to drug substance, it's kind of like, what's that amount you really need to show uh, you're delivering an mRNA, you want to see a protein expression, is it really happening? So for drug substance potency, most of the time it's a characterization assay. You just want to know, okay, is it doing the job? But when it comes to drug product, which is the final product, which is kind of like really translating into clinical trials, you need to have like a more like a release assay, which is always kind of more like a cell-based assay. That's what we most of the viral vectors have done, and like cell therapy has done. So most of the time it will be a cell-based assay. So the strength is kind of more related to the mRNA content. So whatever you're packing into the LNP is kind of like the content, like what is the ideal. So when you look at like a COVID Spivac's lipid nanoparticle, they claim like 2.8 mRNA per lipid nanoparticle has shown like a great efficacy. So these are things we need to understand to really, how much amount of mRNA can be packed, is the size of the mRNA, length of the mRNA playing a role in packaging. So these can be like identified through a regular RT-qPCR, ddPCR, or a reverse phase LC. And the other thing would be like, what's the ratio required to, so exactly like how much amount of RNA is actually encapsulated per particle. Like in content, it's like more related to like, I would say like in ratio, it's kind of like how many nanoparticles are formed and what is that ratio of like how many particles have like one mRNA in LNP, or like how many particles have like more than one. So that's kind of the ratio, RNA ratio to the lipid nanoparticle. So this normally is identified through a digital PCR, which you ideally can say like when you really look at from a viral vector point of view, it can say a copy vector, a copy number. So that's what we are really looking at. So moving on and talking about like a process related impurities. So in this case, like the process related impurities are really that are coming from the process. So the process itself is like, you're generating, you have a plasmid DNA, you're going to generate mRNA, and for that you're going to use enzymes, you're going to use dNTPs, and you're going to use different solvents. So that's the whole point here. So that's what we're really looking at here. So for residual DNA templates, so the potential, the importance, right? Like why do you don't want to have them? So ideally, high immunogenicity activity can be caused, oncogenicity, infectivity, and immunodilution can be caused. So normally a simple qPCR can be done, and it's a release assay. So for dNTPs, normally excess dNTPs can be identified through an exchange, and it's a characterization assay. It's not so. You just want to understand how much are really is everything removed. And residual enzymes, which is like your T7 transcriptases, like to really convert the DNA into RNA, you can just use an ELISA and characterize it. So solvents, like regular solvents, we ideally look at like which can cause toxicity and carcinogenicity. Normally these are through a gas chromatography. That's what we dry them out, and we really look at through a gas chromatography. And lipid related downstream DP product impurities are like, that these are drug product impurities that are based on the lipids. So we need to understand, like, as we are generating these lipid nanoparticles, some of these lipids may not actually form the lipid nanoparticles, but like they can become like more impurities. Again, these are more LC based methods, like again these are very sensitive method, ELSDs, CAD, and MS. And a double stranded RNA is also considered to be like a as you produce more RNA, double-strand RNA can be formed. So that can be like identified through a antibody-based method. So the next method ideally we want to really look at would be for the CQA would be a product characterization. So for product characterization is kind of like you have your lipid nanoparticle. So we are trying to understand the particle itself. We are not really looking at any of this mRNA, but like what we are really looking at is like, okay, what is this particle once it's formed? How is it really looking like? What is it really holding? And what is the charge? So that's what we are really trying to understand here. So in that case, we look at like the encapsulation efficiency. It's kind of like, is it able to really protect the mRNA from degradation, stabilization? So normally we use like a fluorescence based method. Kind of look at like you have an encapsulated one, you have you break open everything, and most of the time we use like a ribogreen assay to really understand how much is encapsulated. A surface charge is an important thing. So normally the surface charge happens through there are like all these ion interactions happening on the lipid nanoparticles with the solvent, and there is some of the ions that are inside the lipid nanoparticles, and there's a jitter potential that's really has been like developed on the membrane. And the whole point would be like the surface charge will play an important role once these particles are sent into an in vivo delivery. If the charge is different, like it can go interact with like very non-specifically to any of the bloodstream, and the delivery is not more, and it becomes non-specific. So that's another thing we need to understand. So it's a characterization assay really to understand. And lipid nanoparticle size. So people have been trying to understand this in a very critical way. So normally people claim that like this lipid nanoparticle size from 60 to 100 is ideal, but there are like people have shown that like around like 40 to 50 is also plays an important role. But like normally these size is identified through a dynamic light scattering or a nanosite. You can also use like a MALS method to really understand that, and this is a very important critical quality attribute to further release. And LNP polydispersity. So we want to understand like if you have like, okay, how many particles are like polydispersed, is like, okay, how many LNPs are around like 50 nanometers, how many are around 100 nanometers, what is the population, what's the polydispersity of this whole product. So again, that's we can use a DLS to really define that. And the lipid content, again, this is completely related to, we've been talking a little bit about this, but from the product point of view, we have a specific lipid content. So normally we have like an ionizable lipid and a cholesterol. So we just want to understand like, are we, whatever lipids we want to form this lipid nanoparticle, is the same lipid really present with the particle. So you ideally go ahead, use like an LC based methods. Again, ELSD and CAD are pretty much used in these things, and these are all like, again, release methods, so they have to be really they have to be really defined with all the LOQs, LODs, and like your range of the method, everything very pretty defined. So looking at safety is kind of more like regular bioburden, endotoxin and sterility. Regular compendial methods have been used, and these are more defined. These are being defined for like a lot of products, and there's nothing special about these things. We just follow the regular norms. Then you go look at the physical properties. In case of physical properties, you ideally look at like appearance. So you don't want them to be like, it has to be clear, transparent, like, and then you also look at like, to really be, yes, if you have a lifeless product, you want to really see that like it's not degraded, like what's the moisture content. Osmolality is an important thing to really check, like, to really, if you want to use it in the injections, like, is it really maintains osmolality. Subvisual particles has recently come up, and like there are new techniques to really understand that. And pH is another thing. What's the pHu? Again, physical properties are pretty much regular, and these all are like more compendial methods. They've been established for a long time, and this is nothing special. we most of the other biologics have the same kind of like testing, and we follow the same testing here. So coming back, like, we have looked at all these different methods. Uh, but what is the challenge of really understanding this critical quality attributes for an mRNA therapeutic? Is kind of like it's just limited knowledge you have, because like people try to make this mRNA lipid therapeutics based on uh, different ionizable lipids, very specific. They want to have their patents. So this limited knowledge. You create this new structure, new things, and you really don't know like how it's going to really play. Um, and again, there is information available, not sufficient information available outside there. As this field has been like evolving from the last 2 years, and it's just still people trying to explore a lot of things. Product development knowledge is limited. We know how to manufacture it, but like really, once it's manufactured, how does that really going to work, or like how to develop that product into a complete final product is something still limited. People are still trying to understand all the other critical quality attributes which can be attributed to this product. That's one thing. And again, regulatory guidance is still evolving. So that's another important point. As we go forward, like regulations will come, will provide more clear clarity how to look at these particles. Here are some of the references to really go over these. Uh, Again, so this is just like a educational talk. Just want to give like what kind of techniques can be like applied. Again, really talking from a critical quality attribute point of view, so it's much easier to connect what kind of techniques can be applied. So that's the whole goal of this talk. Um, and these are some of the references for that. And I would like to acknowledge my kind of like my professional place and my educational place. And I would like to thank everybody for your time, and thank you.